And yeah, so I'm Claire. I'm a technical consultant at Wolfram, and I'll be going over a sort of overview of the Wolfram language and how it can be used for medical research. So the goal is to sort of impart how Mathematica is a complete, highly automated and integrated platform for medical science computation. So we're going to go over what I mean by each of those things as we go through the presentation. So what I mean by complete, highly automated and integrated. So what I mean by complete is something that has a tool for everything you might need from the beginning of your, making your hypothesis to getting the research done, to trialing your sort of theories, to actually then finding your solutions and presenting them to others. So it has tools for every one of those steps. We have a huge amount of inbuilt data that is already in the Wolfram language. We have some pre-trained neural nets. We have here's just a little um, glimpse of some of the things we have. So we have diseases and medical tests, we have machine learning algorithms, and an easy way to do that just in one step. We have image processing stuff, we have lots of anatomy knowledge, lots of medical knowledge, even things for different animals, sort of species knowledge, genes, all these kind of things. We're going to go over a few of them as we go on. So this is just an example. So for some of our knowledge is to do with gene sequencing. So we have a lot of the gene sequences of various different species. So we have the human genome as well. But what this function does is you can put in a string of sort of a DNA sequence, and you can make it into an object. And then with the object, you can do some analysis on it. So you can translate it and into a peptide using this biosequence translate. You can then try and see it as a whole molecule and how it would actually fit together. And you can visualize that molecule because in the Wolfram language, it understands this in every different form. So even though it's displaying in this way, so you can add this function to this is the same function here, and it's looking at the same information, but it's just being displayed in a different way. So that's the thing with the Wolfram language, that it's symbolic, but it also so this is an image, of course, but the Wolfram language understands everything within that image. So that's why you can do sort of multiple different types of, call different functions on it with the same thing to have different results, because it does actually understand, what each of these molecules are and how they're bonded and things like that. So that's why you can transform this object here, which has a bit of information into this sort of 3D image that you can then display and look at and analyze for different reasons. So one of the other cool things that we can do is we can take a DNA sequence for something else. So here it's looking at the gene for this coagulation factor 5. And then we can get that into this sort of object, which will have the DNA sequence. You can see here it's pretty long, 74,578 letters. And then we can use this longest common subsequence, which can be used for other type of pattern matching. But in this case, we're going to use it for sort of comparing genes together. And what it will do is it will look through this whole DNA sequence, and the DNA sequence that we defined up here, and it's going to compare them and see if there are any overlaps, and out of those, which is the longest common subsequence. So they'll both have this same sequence in their DNA sequence strings. So we have all sorts of other information as well. So then this is all inbuilt into the Wolfram language. You don't have to download any additional libraries or anything like that. You can obviously call from places on the internet if you have, using an API. We have API functionality if you already have a database or something like that. But these are all things that are actually inbuilt within the language, so you don't have to go anywhere else for this. And there are advantages to that, because this can then be part of an entity which holds lots of different information around this thing. So here, for example, we've got this entity of the anatomical structure of the heart, and this will display it in this nice way. But if you're not familiar with what an entity is in the Wolfram language, it's basically an object that holds a lot of information about a particular sort of subject. So in this case, it'd be the heart, and so it will understand all the different anatomical features of the heart. So even though here it's displaying it in this one way, you can actually go further within that, and you can say, oh, I actually want this valve to be this color, and I want the rest to be this different type of opacity. So that way it can be kind of see-through, and then you can have the bits that you actually just want to see visible like that. So it can be really nice to visualize things and how understand how it looks in a sort of 3D way. So you can take that and then again sort of use the same entity for that. And we'll look a little bit more about entities later. But it's basically, it'll be the one main thing, and then it'll have all these different properties. So you can look at various different bits of information that we actually hold on that entity. It can also be good to actually augment your own data. So if you've got a database and it returns something, like a heart or a different anatomical feature, you can sort of augment your data by turning that into a mathematical entity. And then you also hold all the other information we have around that topic. So I also said we have some medical testing data. So you can, this is a sort of entity, and you can find all the different ones that we have. I think by just doing this. So these are the different medical test data that we have. So yeah, you can just have a look at those if those are useful to you. And you can get various things with that. So again, these are entities, which means they have different properties. And so here we've taken the mean, median, and reference range of 90, and we've just taken those properties from the serum iron medical test. And we can take that data and we can display it in different ways. So here we're saying I want the basic metabolic panel, and I want to sort of display it in these different tables, a distribution plot with the name here, and then the medium displayed, the mean displayed here. So you can sort of compare the different tests that we have and do sort of analysis or comparative things that you want with that. Okay, I also said that it was an automated platform. So what I mean by that is that a lot of in the Wolfram language, it's attempted, if you really know your subject, you can make things a little bit more specific. So, we're going to go over some machine learning stuff, and you can add your own methods. You can say, I want it to use, particularly binary tree or logistic regression, or, you can say specifically that you want it to do that. But overall, the Wolfram language tries to figure it out itself and allow you to not have a full extent of knowledge of how to do every single thing and be a specialist in everything. It will try and figure it out for you, and sort of allow you to put as little input in as possible. So we see that in a lot of places within the various functions that we have. So here I'm going to look at our machine learning, so a neural net making data. So here I'm looking at this mitosis data. It takes quite a while to get this data and to classify it. So this is actually how you call the function. I'm not going to call it now because it takes several minutes, but if you get this notebook later, just remove these comments here, and you can run that if you want to sort of train your own neural net. But before I get to that, I'll just show what this mitosis data is. So this is a list. I think it's about 2,350 something pictures long. So that's how many bits of data you're getting. And this is basically what the data is. So it's an association here, and it's got an image, and whether it was it had a whether it's suspected to have a tumor or not. So if it had a tumor in the photo, then it would say have this picture, it goes to true, and if it didn't, it would have the picture and it will say it goes to false. Um, Obviously, this is quite a small sample, so as we'll see, it's not going to do amazing, because to do a study like this, particularly with things that they don't look all that different from one another, a true and a false. I mean, I find it difficult to tell specifically myself looking at it. Obviously, I'm not an expert. I'm sure someone who was trained in it could. Um, but for a machine learning algorithm, obviously you need quite a lot of data. So to train it, first we're going to get this data and we're going to split it so we get so we can have some testing data, that the machine learning algorithm hasn't seen, because obviously there's no point using data that the machine learning algorithm has already seen to test it, because it's going to do much better and give you not very accurate results, because it's already seen the data, so it already knows how to classify it. So generally, once you've got this data, so we've saved this into training data by just taking a selection of the full mitosis data. And then what we would usually do is we would put classify around it. And what that does is that it looks through all the data, and again, you don't have to put anything else. This is literally the full function, and it will figure out what the best method machine learning algorithm is to use, it will figure out that there are sort of these two categories, true and false. You don't have to put any of this extra sort of features in, and it will try and classify that based on, we're not sure what exactly. It's always hard to tell exactly what features it's using. There is some stuff where you can have a little bit of a look inside, and it can give you a little bit of feedback on how it's making its decisions, but we won't go over that today. So like I said, I'm not going to run this classifier function live because it would take quite a long time, particularly on a webinar, because it sort of takes a lot of my computing power. So I'm just going to get one that I've already done, but I did it just the same way as here. And then what this returns to you is this classifier function, which allows you to put in new data, and then it will try and classify it in this case whether it's true or false. And you can see it's used the method of logistic regression, and that's the number of training examples it's used because the rest of it was preserved as reserved, sorry, as testing data. Um, so then we can get some information on how it was run. So this sort of says just a little bit more information than is up here. So it says the classes are true and false, how accurate it thinks it was, the method, logistic regression, how long it took, those sort of things. So if you're interested in the data and how it chose its thing, you can have a look at this there. There's different graphs that you can click through as well. Um, so then we can take this data and test it with our training set. And here in this random sample of 10, it's actually done very well. It's correctly classified all of these images that did not show signs of a tumor as false, and it did correctly do that. I think there were more images that were false than there were true. There we go. Okay, so here I've just run it again to get a different random sample, and you can see here it's correctly identified it as false. So this is what the machine learning algorithm classified it as, and this is what it is actually, according to the according to the data. So you can see it's done all of these correctly. This one it misidentified as true, even though it was actually false. It didn't have a tumor, didn't have signs of a tumor. These were all correct, and this one as well were correctly identified as, uh, potentially having a tumor, whereas the other ones didn't. So that's done pretty well. And yeah, again, that's just taken the random sample of the testing data here, and it's gone through basically each of those things and put it into this mitosis queue, which is the variable we used for the classifier function that we got earlier. So then we can look at that, and we can look at data from that as well. So how it's done on the training data, it will tell you how accurate it was on that data. Again, obviously, it's going to be the same method, and this is sort of really useful. So this will tell you what it did correctly. So you can see that 455 examples of false were correctly identified as false. However, 19 images that were false were misidentified as true. And this is where, you get something that you really don't want. What you really want is these two boxes to be, the highest, and then these to be zeros in an ideal world, which would mean it didn't misclassify anything. But you can see it had a little bit of trouble here identifying what was true or not. So it had, 50 examples that were true. It actually misidentified or misclassified as false. Um, and then these are the ones it got right. So 64 examples of true were correctly classified. And this can be really useful because it shows you where the data was perhaps lacking. Maybe there weren't, and you can see there weren't as many images that did actually show signs of a tumor. So maybe there should be more examples of that. So the neural net can have a little bit more to go on, have a little bit more information, and could get more accurate. So that should really sort of identify to you that it needs more examples of that. Because with the false, although it did get 19 examples wrong, and it did misclassify it as true, which isn't as bad, obviously, it actually did identify correctly quite a lot of them. So it sort of gives you some sort of key on that, or sort of idea on that. You can also look at SHAP values. Again, we're not going to go over that today, but we do have the possibility to look at SHAP values, which, if you're not aware, it basically, if you've got something that takes a lot of different values in, so obviously in this case, it's not just a true or false of the same image, but if you've got something that takes 10 different variables to classify one thing, then you can look at SHAP values, and it will tell you, which of those those different inputs were used more or less or had more of an impact on the final result, which can also be really useful if you're looking into machine learning algorithms. So this is just to show that you can actually pick out specific things within this. So you don't just have to get this whole table. You can sort of pick out just this little diagram, or you can just look at the accuracy, this specificity, how accurate it was in true or false, and the sort of error rate and things like that. So you can pick out exactly what you need if you don't want to sort of get this whole table. Okay, and then I also said that it was an integrated platform. So what I mean by that is that we have the ability to do lots of different type of analysis. So we can do image analysis as well as looking at graphs and mathematics, and we can quite seamlessly go between those different things. So I think we've had examples of that throughout the whole thing because it's a big part of Mathematica that because it's also symbolic and behind the scenes, it's all very much the same. So everything has this sort of function name with the square brackets and the things inside. So broken down behind the scenes, Mathematica is very similar. So that means that you can really um, it's very easy to use sort of one bit of data in multiple different functions because it sort of knows how to work with that because behind the scenes they're all very similar. Even an image behind the scenes, it's just a sort of a lists of different numbers and pixel values and things like that. So I'm just going to make this a little bigger. So this is an image of stained cells from a rat brain, which some of them have sort of more of a fluorescent uptake than others, with this sort of green, as you can see, and it's sort of split into this blue and green uh, from the staining of the cells. And so we can get that image, and that you can just drag and drop an image in, or you can sort of import one. There are different ways to get images in, but this is a notebook, and you can just drag and drop. We can then actually color separate this image. So Mathematica understands what red, green, and blue are, and you can separate uh, this image. Actually, you'll know it's not actually using the predefined values of red, green, and blue. We're actually putting that on ourselves. That's just a variable name, and we're using this function color separate, which can actually separate the image based on that colors. So here we're gonna put a bilateral filter on it and make it all this kind of uh, sort of gray, and then this morphological binarize, what it does, it makes everything either a 1 or a 0 basically, so that's why it's binarized. So either it's white or it's black, and that's why you can get this really clear contrast, which can be useful to do further analysis. So now we've split this image into very clear sort of partitions in a way. We can do some other things a little bit easier. So here we're going to use this filter to try and get the center of each of those things. So basically it kind of takes the whole thing where there's differences and tries to go in and in till it gets to sort of midpoint in the middle. Um, and so it kind of looks like some stars now, but this is the center of all the different cells. So this can be useful again because then we basically overlay that over this the original sort of black and the grayed image here, and we can see the points in the middle where that was sort of overlaid on top of it and made pink, and it's all highlighted here. So you can see that that's done quite well, even these that were quite close together, and these ones you can see they were differentiated as two separate cells, and you've got different points in each of those, um, and you can count those and things like that, and you can partition it. So here, just to make it easier, we've filled in every cell that it's identified with a different color, um, using these sort of various functions sort of put together, and we can get these different components. And again, you will be given this notebook, so you can, go further on this and do your own analysis, use your own images and things like that if you want. And then we can go straight from this sort of image analysis to sort of doing all these different things to putting those data points into a histogram very easily. So you just use histogram and you put the component measurements of the things we've literally just done. With the green and the components, we're looking at the mean, and we can put it into a histogram without any problem. We can then use this manipulate function, which doesn't even need to be told to put it into a slider. Sometimes it will just look at your data, and you say what you want to manipulate, and then it will figure out a way to manipulate it in a sort of sensible way. So in this case, it's chosen a slider, and what we're looking at is we're looking at what has more fluorescent uptake. So as you go sort of down, you have the ones that didn't have a lot, and you can see sort of all of them quite faintly. And as you go up, you get the ones that were sort of really bright. Um, and that's probably the brightest one. So then you can go up and down, and you can sort of see the different collections and how the different ones were reacted differently to the fluorescent thing, or were dyed a different color. Um, so that can be quite useful as well. And as you can see, we've sort of gone very easily and used the same variables between the sort of image analysis, and then finding these components of the things, putting it into a histogram, and then actually having an interactive um, thing that you can manipulate that can use those variables and actually sort of give you a different thing again. And so it really works seamlessly with one another. You don't have to have a library for, looking at images. You don't have to have a separate thing for doing graphs. You have a separate thing for doing things that you can manipulate. It's sort of all try to be rolled into one, so you can do everything within the notebook. Is the idea. If you've come up with a cool idea, or a cool research project or something, you can create these things which allow exploration, but in a constrained manner. So very much like the manipulate things, um, but slightly different. So here, for example, we've got the sort of bones of the sort of torso and pelvis, um, and you can highlight various things in it. So here, let's get the atlas, so we can get the top one. Oh, is that coming up? Oh, there we go. So you can sort of highlight that there. We can get the scapula, turn it around. Just going to turn around, and then you can sort of see the different highlighted things. So again, you can get an idea of things in 3D. There are lots of different things like this. So these were actually, if you go, this example was taken from the Wolfram demonstrations page. So if you go to this page, you can get all sorts of different little examples of things that people, users of the Wolfram language, have put together, and they've wanted to share with others. So you can go to this page, and you can run through and look at all the different examples. These were ones that it was biology based, but you can look at various different ones. Here it's looking at the growth of teeth, horns, and claws. You can, there are the temperature changes for inhalant gases. You can get various different things. And also, what's quite good with this, so you can click on it, and if you have Mathematica, you can actually download the source code as well. So that way, if it does something that you think is quite good, but it's not quite what you want, or you want to expand on that, no problem. Just download the source notebook. You can copy and paste this into your own stuff as well. You can adjust things that you want to adjust. And also, obviously, you can just use the tool as it is. I think this one sort of changes color and gives you the temperature. Not exactly sure, but you can read through that, and there'll be explanations, and it's just really useful. So if you find something in there that does something that you quite like, you can actually go in there and alter it. It's the same with resource functions as well. So we have this sort of resource. To find the reference for it. So it's a function repository. So um, I'm sure many of you are already aware of this, but for those of you who aren't, there's the Wolfram function repository, which is sort of additional to our normal built-in notebook. And this will have all sorts of different functions that people have added themselves. So users of the language have added these functions that they think aren't in the built-in language. And again, you can, so I don't know, biosequence graph, looking at more sort of gene analysis things. So you'll see it is very similar to the documentation page. It will give you some details, some examples, the applications. And again, you can download the source notebook, so you can get all the code. It'll be Mathematica code that was written to write this function. Um, you can copy and paste this into your own notebook and alter things if you want, or you can just use resource function. You just type resource function and the name of this, and then you can just use it like a normal function without having to download or copy and paste any code or anything like that. So yeah, definitely go and look at the Wolfram demonstrations page. Lots of stuff on there, it could be useful. So in conclusion, I hope that I've shown how the Wolfram language tries to be a complete and highly automated integrated platform for medical science. So thank you for listening.